got the TB blues.
So you were just listening to Jimmy Rogers sing the TB Blues, and Jimmy Rogers was a bluegrass singer from just at the early part of the 20th century. And I do collect a lot of songs about diseases. You will hear a lot of them, yes, of course. Um, but this one is a very serious song. Most of the songs that I will play are very lighthearted and novelty songs. But Jimmy Rogers actually did die at the age of 35 from tuberculosis. So, why did I have this particular song to begin this lecture? Well, we are going to talk about a lot of unique types of bacteria, and we are going to talk about tuberculosis. Okay? So, yes, it is relevant. Didn't mean to bring you down. Let's come back up again. So, where are we? So, this is the week of October 23rd, creeping towards Halloween, right? And this week it's all about cells. Now I know your attention is drawn to the right. I just wanted to mention now these are our three SIs and helpers in the laboratory. Maybe you know these gentlemen. If you've been on campus, you probably have seen at least one, if not two of them. But from left to right, that's Jeremiah, and then Eustace in the middle, and Andy. Now, Eustace is not very useful in the laboratory. So, anyway, that's our Halloween Eustace. If you stop into the laboratory just to say hi before Halloween, you'll be able to see Eustace. And maybe you'll run into Jeremiah and Andy. Okay, so what are we doing this week? What are the objectives? The objectives that I gave you in some respects were overambitious, so I took out the last three. I think what we're going to do is we're going to talk about prions and viruses in the next lecture. I don't think you will mind this, right? Because I thought we had enough to talk about. I will touch on viruses, however. One objective that I did not include when preparing my section of the syllabus months ago was the compare and contrast gram-positive, gram-negative, acid-fast, and atypical bacterial cells. So that was an addition. Notice I also have that highlighted in red. So a red font in a bold type. I did that not only because I wanted to draw your attention to an objective that I forgot, I wanted to draw your attention to this because this is a really big, important aspect of this lecture. The weekly assignment, what is that going to be? Well, that is going to have you finding a short video online that you feel is most useful in demonstrating how your bacterial pathogen infects and lives in the human host. And basically, I want you to look for host bacterial interactions in that in your video. So if that's at all possible. So you're going to find this great video and you're going to post the address, the link, so that your fellow classmates can also see that video. The instructions will be online as always. Okay. All right. And here's some terms, and I went through here, took a lot out, added a few things, so hopefully it's okay. Oh, look, I have some other things that are in red and in bold font. These tend to be really important, and in that, they're going to be important themes throughout the semester. Okay? All right. We are going to talk about certain kinds of bacteria, and as far as these bacteria are good representatives of certain bacterial types. So I will bring these bacteria up in the lecture. That also means that you will be responsible for knowing that this organism has this particular bacterial characteristic. So, for example, Bacillus anthracis. We're going to talk about that. Yes, it causes anthrax. And it is something called gram-positive, and it makes endospores. I have Clostridium up here. I have E. coli. I have Mycobacterium tuberculosis. Yeah. Mycoplasmas, rickettsias, staph and strep and a few others. So if I mention a certain bacterial type as representing the type of bacteria that have a characteristic, like a cell characteristic, morphological characteristic, then you probably need to know that. All right? All right, so let's start with that big time objective. Compare and contrast prokaryotic and eukaryotic cells. I'm going to begin with a brief comparison and then we're going to do some detail work, okay? All right. So before we do that, just note that both prokaryotic cells and eukaryotics are pretty small, and under the microscope, we tend to measure them in units of micrometers or microns. So this is the Greek letter mu, right, and the M for meter, and that's typically the size unit that we use when measuring cells. Not always, but usually, okay? So, prokaryotic cells versus eukaryotic cells. What are some differences here? First, let's look at the prokaryotic cell type. Now, if an organism is has a prokaryotic cell characteristic, then we call them prokaryotes. Okay? So, what does prokaryote mean? Well, whenever you see kary, K-A-Y, K-A-R-Y, that usually it has to do with a Latin word, and it means nucleus, okay? Pro is always kind of before or pre, so prokaryote means pre-nucleus. These cells, they were around before a nucleus was seen in a cell, so they're pre-nucleus cells. Now, they're still around, clearly, but there you go. All right, so how about an example of a prokaryotic cell? Well, bacteria. Bacteria are prokaryotic. And here's a little cartoon of a prokaryotic cell. We'll talk about this in some detail now. So some general characteristics of all prokaryotes, and that would mean general characteristics of bacteria. Okay, link them together. Bacteria, prokaryotes. Okay, now, of course, there are other prokaryotes. Those are the archaeans, but we're only interested in bacteria. Okay, general characteristics. Prokaryotic bacteria tend to be very small, sometimes maybe a single micrometer or maybe 2 micrometers. Very small, very simple, not complex, very simple. Prokaryotic bacterial cells are always unicellular. What does that mean? That means that they are a single-celled living creature. Unicellular, single-celled living creature. They don't have a nucleus, they have a nucleoid area. That is an area in the bacterial cell where the DNA is held. Okay, so it's not a true nucleus, and they only have one circular chromosome, just one. Okay, it's made of DNA just like ours, but it's in a circle. So if you took the DNA ladder that you think of when you think of DNA, and you take the top of the ladder and you twist it and attach it to the bottom of a ladder, you have a circle, and that would kind of look like a prokaryotic bacterial chromosome. Now, bacteria, prokaryotes might have extra round pieces of DNA, but they're not chromosomes. They're not required for the bacterial cell. They usually carry extra pieces of genetic information. These are called plasmids. Bacterial cells, prokaryotic cells, do not have any membrane-enclosed organelles. What is an organelle, you ask? I'm glad you asked. An organelle is any structure in a cell with a function. So, an organelle could be defined in slang terms as a cell thingy, a thing that does something. So that's pretty much everything. Okay, so let's compare and contrast that to a eukaryotic cell. Now, if you are an organism that has eukaryotic cells, you are considered a eukaryote. Okay, so these are the eukaryotes we're talking about. So eukaryote, the capital E and U here, okay, EU always means true. Like in the last lecture, I said that eubacteria mean true bacteria. Well, eukaryote means a true nucleus. These cells have a true nucleus. Okay, examples. Well, up here is a human neuron, a nerve cell. Okay, you've got a lot of these. So you have your peripheral nerves and you have your central nervous system, right? So the human nerve cell illustration is on the right, but there are other eukaryotic cells out there, and some of these we're going to talk about because they are medically significant. So fungal cells are eukaryotic. Protozoans like amoeba and a few others like flagellates and ciliates and apicomplexans, that would be the protozoans that cause malaria. We haven't talked about those, but we will. Okay, those are all eukaryotic. Some general characteristics. Okay, eukaryotic cells tend to be considerably larger than a prokaryotic bacterial cell. Now, are they unicellular or multicellular? Yes. Okay. Eukaryotic cells could be, you could be a unicellular creature and eukaryotic, like yeast. Yeast are fungal, they are eukaryotic, but they live their lives as single-celled little creatures. But you can also be a multicellular eukaryotic organism. So that would be like us. I'll tell you what, if you're multicellular, there's no question you have to be eukaryotic there, but unicellular, though, is not unique to just bacteria. Okay, like I said, eukaryotes can also be unicellular creatures. Eukaryotic cells tend to be very complex. They have a true nucleus, like we already mentioned before. We have more than one chromosome. We have many chromosomes, and they are linear, like a twisted ladder. They're not in a circle, they're linear, like a line. The eukaryotic cells have a lot of membrane enclosed organelles. Yes, so lots of structures. When you have a lot of stuff, you got to put them in containers. And if you're a cell, what kind of container can you make? Well, cells make all those phospholipid membranes, so why not wrap a lot of things up in those membranes? And so you can have membrane enclosed organelles. All right. So here is a microscope picture. And what are these things on the microscope slide? I'm glad you asked. So the blue guys here are neutrophils, and they are infected by the pink bacterial cells. Now, these bacterial cells cause Rocky Mountain spotted fever. So we call, by the way, the bacterial cells in pink are their species name is Rickettsia rickettsii. And like I said, they cause Rocky Mountain spotted fever. So notice the size difference. So the purple cells are, now they're inside our cell here. So this is a white blood cell known as a neutrophil in blue. And the pink cell is a prokaryotic bacterial cell. Yeah, big size difference here. So again, the blue neutrophil is a white blood cell, and it is much more complex. It's a little hard to distinguish the membrane enclosed organelles, but they're there, and you can kind of make out some of this. They have a true nucleus that you can see. Oh, yes, membrane enclosed organelles. Oh, yes. Now, the red kind of pinkish bacteria are much smaller. They're much more simple. There's no true nucleus. Their round circular DNA is just wrapped up in the nucleoid area, although you really can't see it in this picture. They do have organelles, but they're not wrapped up in membranes. And again, you really wouldn't be able to see that in this particular picture. All right. So how about a little quiz? Okay. Which cell type has a true nucleus? A prokaryotic, a eukaryotic, both or neither? Right, so prokaryotic means the cell, this is pre-nucleus. They don't have a true nucleus. Eukaryotic, true nucleus. So the answer is B, eukaryotic. Okay, I have this here, by the way. You must watch the lecture for the answer. some students, they get in a hurry and they don't want to watch the lectures and they try to read over the slides. This is to encourage you to listen to the lectures because I have some wonderful gems to share with you. So your life is going to be so much easier. You're going to learn so much more if you listen to the lectures. Okay? All right. So this one's a trick question. Which cell type has organelles? Prokaryotes, eukaryotes, both or neither? Did you say both? I said an organelle is any structure in a cell that has a function. Both prokaryotes and eukaryotes have organelles, but the eukaryotes have their organelles wrapped up in membranes. See the difference? I know sometimes it's not very clear about that, but an organelle is any structure in a cell that has a function. Okay, cell thingy. Hey, which cell type has DNA? Prokaryotes, eukaryotes, both or neither? Yeah, absolutely both. You can't have a cell without some kind of genetic information. It's just that prokaryotes have a single circular chromosome. Eukaryotes have multiple linear chromosomes. But all those chromosomes are made of DNA. Okay? So you're doing your project right now, right? You have to write your outline and you have to write your research paper. So that bacterial organism that you're writing a report on, what would that be? Prokaryotic, eukaryotic, both or neither? Well, it's bacterial, so what does that have to be? Right, prokaryotic. You got it. So your bacterial organism is prokaryotic. So all the characteristics that I cover, the morphological characteristics of prokaryotic bacterial cells, use that when writing up your research paper. Okay? Right. Connect the lecture material to your project. That's the whole point, you guys. So today I'm going to be giving you an awful lot of information that you can use in your research paper. All right? Make sure you cite me. And you can go on and find out how APA suggests that you do that. Still use other sources, but you can cite me. You betcha. All right. So let's go ahead, identify and describe prokaryotic bacterial cell structures. Now we're going to get into some details. If you thought that was it, no, we have some big details. But there's a reason that we want to go into the details. And I will tell you, so another instructor that has taught microbiology said, oh, everybody tunes out when we start to talk about cell structures, eukaryotic versus prokaryotic cell structures. And I thought about that and thought about that, and I thought, it's because the students don't get why they have to know about the cell structures and why the difference is important. So I'm gonna tell you why you have to learn these structures, and I'm gonna do you a favor. Okay, if a cell structure or function is not really all that medically significant, I'm not going to cover it unless it is something really basic, okay, and that you kind of have to learn that to set up a foundation. But I'm going to do you that favor, okay? All right. You say, it's a deal, Miss Eden. Okay, we're good. So let's start with shapes and arrangements, okay? So we're going to talk about bacterial, prokaryotic cell shapes and arrangements. Some of you already know the shape and arrangement of your particular bacterial pathogen that you've been doing your report on. So let's see if you've got it. All right, morphological characteristics of prokaryotic bacterial cells, shapes. The way I have the slides set up is I always have the main topic here highlighted in red, for the most part. Okay, there are tons of versions of bacterial shapes, by the way. I'm just going to cover the basics. First of all, something that looks like this, kind of rod-shaped, is known as a bacillus shape. Bacillus is singular, bacilli is plural. Okay, bacilli is plural, bacillus is singular. So they're rod-shaped. And they could be short rods, long rods, thin or kind of fat, but they're rod-shaped, okay? Now, some bacteria are even named after their shape. So, for example, Bacillus anthracis causes anthrax. Guess what shape it is? You got it. All right. So, when scientists were coming up with the name, they looked at the bacterial cell shape and they said, well, it's bacillus in shape. Let's call it Bacillus anthracis. Okay. So to the right here, we see these are cocci, or singular is coccus shape. And so that would be spherical, wouldn't it? Yes. Some bacteria are named after their shape as well. So for example, Streptococcus pyogenes and Staphylococcus aureus. Strep and staph both have a coccus shape. So yeah, they threw in that coccus name. They threw that into the genus name of their organism. So how nice. So you look at the name and you know, oh gosh, that must be a coccus shape. What's the streptostaphylo stuff? We'll get there. Okay, there's also Spirilla or Spirillum, is singular, and there's several different kinds of shapes of Spirillae. But again, they're kind of spirally. Some of them are flexible, some not. There's a special kind of spirillum, and these are known as spirochetes. They are coily and flexible, and this would be a good cell shape for those of you who are doing your research papers on, oh, I don't know, Lyme disease, Borrelia burgdorferi, or if you were thinking about syphilis, Treponema pallidum, those are spirochetes. Okay. There's also something called a sorry, that wasn't supposed to come up like this. Sorry, coccobacillus. This means, golly, it's such a short bacillus, it almost looks like a coccus. So they threw it together and called it a coccobacillus. So it's a very short bacillus shape. One other shape is called pleomorphic. That's the term scientists use when they don't know what to call it. Some bacteria and other things form various shapes just depending on what day it is, where you find them. So we call this pleo, meaning many, morphic, meaning shapes. Okay? Or characteristic types. Okay, so here over here, yeah, it's like, well, coccus, no, kind of what is that? Okay, so we'll just say pleomorphic. We'll find out that there are viruses that are pleomorphic as well. Okay. Oh, okay, so what about arrangements? Yeah, there are many different kinds of bacterial arrangements. So some bacteria are all by their little lonesome selves, and we say they're in a single arrangement. Sometimes they are diplo. Okay, so this is a single coccus, and these are diplococci. Sometimes they are in a strepto arrangement, like these streptococci. Sometimes they look like they are in clumps, like grape clusters. We call that a staphylo arrangement, like staphylococci. Now, certain things are probably triggering in your head, going, okay, I've heard these terms before, I think. Yes, some bacteria are also named after their arrangements. So, for example, Streptococcus pyogenes group A strep, like that causes strep throat. Yeah, strepto arrangement. Okay, Staphylococcus aureus. Yes, so they are in a staphylo arrangement. You're like, I'm getting this. Good for you. Okay, good. Hang in there. Now, sometimes, though, just when you think you've got it, sometimes the scientist who names the bacterial organism will give it a deceiving name. And it's not my fault. Okay, I just want to say this. So, for example, Streptococcus pneumonia. You're thinking, yay, it would look just like this, but no, it's actually a diplococcus. Sorry, not my fault. You'd think they'd change it after all these years, but no. Streptococcus pneumonia is actually a diplococcus. This is the organism most of you have in the back of your nasopharynx, and it causes pneumonia in a lot of your patients. A lot of your patients at the very end die of pneumonia because of this guy here. Okay. Okay, and here they are. You're like, wow, that's small. I know. Try looking at these under a microscope. But here's one, oh, here's two little round cells, Diplococcus. And they're making this very clear zone too. We're going to talk about those. Those are capsules. That's coming up. Makes them very, 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 very resistant to all kinds of things, like being killed by our white blood cells. So that's a Diplococcus right there. Now, what about the bacilli shapes? Glad you asked. You know what? They have similar arrangements. They can be in a single or a diplo. They can be strepto, but because of the way they divide, they tend not to be staphylo. The closest they can come to a staphylo is looking like a picket fence and being kind of next to each other. These are called the palisade formation. The bacterium that causes diphtheria, called Corynebacterium diphtheria, has that unique arrangement. You don't need to know this about Corynebacterium diphtheria. I'm just giving you an example, so for your information only. Okay? So, we're going to talk about some bacterial structures now. Like I said, we're going to talk about structures that are relevant medically, going to be relevant to you. Some of the bacterial structures that we will talk about will actually mount an immune response in the host. And some of these structures are targets for antibiotics or antimicrobials. All right. So some structures on bacteria will be identified by our immune systems. We call these structures identified by our immune systems antigens. What is an antigen? How do we define that? An antigen is anything that generates an immune response. Think of them like some kind of ID tag, okay? So the bacterial organism or even a virus or something gets into the body and it looks weird. It's got strange things, proteins and carbs and lipids sticking out of it that is identified by our immune cells. So we would call those all antigens. Okay, anything that generates an immune response. So here's a little E. coli. This is an animation representing E. coli. And you can see there's all kinds of things sticking out there. And yeah, there's a lot of antigens sticking out of E. coli there. Okay. Okay. So let's talk about the structures known as flagella. Some bacteria, not all, have flagella. Oopsie. So I'm going to show this illustration again and again and again. We're going to be looking at structures on the outside and on the inside of the bacterial cell. Okay. All right, so flagellum is singular, flagella is plural. Over on the right, you can see this little bacterial cell has a single flagellum. The rest of them have flagella. Sometimes they come out of one end, sometimes both ends, sometimes they're all over the place. Okay, like E. coli, or this is Pseudomonas aeruginosa down here, and they are covered in flagella. Now, flagella allow for movement, what we call motility. Okay, so here are the flagella, the long fibers coming out of the end. So what is motility with the T? Did she misspell that? No, I do misspell things occasionally, but no, that was spelled right. Motility means that these guys can move under their own power, and they do, where do they have to go? Why are they moving? Well, they move towards good things, like, that looks good, or, fresh air, and away from bad things. And so they pick up chemical signals through their receptors, and they head towards, smells good over here, or blah, smells bad over there. And so they're picking it up through chemical receptors. So that's motility. Now, what about mobility? Now, some bacteria, they're not motile, but they're mobile. So that means that they just float around in the circulatory system or through the lymphatic system, but they're not moving under their own power. So, for example, your patient may not be motile because something happened, maybe they're paralyzed, but they can be mobile. We move them around in a wheelchair. Okay, got the difference? Good. All right. All right, so there are special kinds of flagella that are wrapped around the bacterial cells. These are called paraplasmic flagella. They're only found in spirochetes. Remember the spirochetes? Yeah, they're corkscrew-like. They're flexible. Okay, they're kind of a spirilla shape, a spirillum shape, sorry. So they have these filaments that wrap around sort of like a ribbon, and they spiral around the cell, and they move against themselves, and they cause the cell to flip and to move. And so an example would be, oh, Treponema pallidum that causes syphilis. So here we have an example. Now, if you're thinking, I would like to see these move. Well, I'm here to answer. Oh, no, no, no. Oh, well, let's put up the other example. Another example would be Borrelia burgdorferi that causes Lyme disease. Okay. No, 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 no. Not yet. Okay, stop it. Okay, so here we go. Now you can see how they're moving in a corkscrew kind of manner. Wow, would that penetrate tissue really well? What do you think? And there they are. And you can kind of see the flagella filaments and how they're wrapped around like little ribbons. Just imagine how if these guys got in by sexual contact, like syphilis, they'd move very quickly. Or if they were injected by the bite of a tick, and these were Borrelia burgdorferi that causes Lyme disease, where could they go? Pretty much everywhere. So, little bit about Treponema pallidum that causes syphilis. They move through the skin, down through the basement membrane, into the blood vessels, and they can do this very, very quickly. So they move so well and so fast with that spiral movement that they can get to and invade most all tissue, including the brain, within 48 hours after entering the host. So just think about that. And this is probably pretty much true for Borrelia burgdorferi that causes Lyme disease. Is that going to cause a huge inflammatory response by your immune system? Is your immune system going to like this? Probably not. So you might have some inflammatory damage that might occur. But this is why these spirochetes are medically significant and why I wanted you to know about these. Okay? So notice they're in red. Okay. All right, let's look at another structure. And these structures stick out of the bacterial cells as well, but they're not flagella. These are known as pili. Not all bacteria have pili, just like not all bacteria have flagella, but a few do. Pili are mostly found in gram-negative bacterial cells. You're wondering what that is? I'll tell you later on, but that was just a side note. Don't worry about it now. So, okay, so pili is plural, pilus is singular. So here we have these little structures sticking out here. What are these? Well, they have a couple of functions. Now, sometimes bacteria have these pili and they allow bacteria to pass DNA from one cell to another. These are called the sex pili, or it's a sex pilus. So this is for bacterial sex. Okay, and over here, these are cartoons that give you an idea of how this happens. So here we have a bacterial cell, and here's another bacterial cells, and they meet and they care about each other very much, and so they're about to have bacterial sex. So here in blue is the bacterial chromosome. These in orange and yellow, these would be the plasmids. Remember, these are extra pieces of genetic material, not part of the chromosome. But using the sex pilus here, this bacterial cell, okay, can attach to the other bacterial cell and send genetic information across the sex pilus. Now, this is important because the genes on these pieces of DNA, okay, they could contain genetic information to make toxins. Maybe this bacterial cell didn't know how to make a toxin before, but this one did. And so it sent over the information. Or maybe the genetic information is, this cell has the genes to become antibiotic resistant. So this is a superbug. I use the B word, I know. This is a superbug. This one isn't. This one would respond to antibiotics. But over this red plasmid's passed over, so now they're both superbugs. Okay? All right. The pili can also be used for attachment, allowing bacteria like E. coli to attach to all kinds of things, not only to not only to the host like cell surface. So, for example, E. coli can attach to your intestinal cells sometimes. Okay. But if E. coli that originates in fecal matter get in the water, and then you water your vegetables with feces that you can't see, but E. coli is in that fecal material, and you water your plants, and E. coli can very easily attach to the produce, and then you try to wash it off, and they won't wash off because they're holding on very tightly with these pili. All right. So that's one of the reasons why sprouts, that's why they sometimes produce infectious diseases due to E. coli. So there's E. coli outbreaks in a lot of vegetables. Even if they're pre-washed, the bacteria like E. coli, they can hold on. This structure also allows E. coli from being swept out of your urinary tract when you urinate. What? If you were okay, so E. coli is an intestinal uh, organism. It lives in animal intestines. You all have E. coli in your intestines, right? We talked about that in the first lecture. But E. coli can accidentally get in the urinary tract and cause urinary tract infections. They can hold on to the urinary tract epithelial cells, like in the bladder, and they hold on even when you urinate. So let's take a look. You're like, what? Yeah. So here's a nice picture of the bladder, and you've got all of these, your bladder is lined with epithelial cells. So here is the histological picture. And E. coli can swim up the urethra and get into the space and attach to your bladder using a sex pilus in black. Yeah. By the way, most urinary tract infections or UTIs are caused by E. coli. I have pictures of cranberries here. Why are there pictures of cranberries here? So why are cranberries possibly beneficial in treating UTIs? You know, how they tell patients when they have urinary tract infections, have some cranberry juice. Why? It has to do with a chemical called proanthocyanidin. Okay, it's a proanthocyanidin chemical. You're like, bless you. Okay, just know it's a chemical in cranberries as well as in other plants. Okay, what that chemical does is that it blocks the pilus. So it sticks on the bottom of the pilus structure of E. coli. And if the bacterial pilus is all blocked, then it cannot attach to the lining of your bladder. And that is why. This doesn't work with all E. coli's, I'm afraid. But it can't hoit. It can't hurt. It's a useful drink. So tell your patients that, yeah, they have to drink their cranberry juice. It has to be made from real cranberries. Okay? Little side note about urinary tract infections, particularly those that are caused by E. coli. Little girls are very susceptible, as well as postmenopausal women. What about the boys? Uncircumcised boys are very susceptible, as well as Caucasians. We don't know why. It's just the way it is. So why is this? Well, sometimes we recommend that babies and women not take baths. Why is this? Well, baby's getting clean, but baby has a lot of fecal material. Okay, it's a baby. And in feces, you're always going to find E. coli. E. coli is motile. They have flagella. They swim. So they can swim from the rectal area up through the urethra. Okay, or, basically they can get to where they need to go. And that is why. Little side note there. You're like, oh. How about another structure? Okay, we're going to talk about another bacterial structure called a glycocalyx. Not all bacteria have a glycocalyx, but some do. We're going to especially talk about the type of glycocalyx known as a capsule, but there are actually two types. So, all right. So glycocalyx, what is this word? Glyco means sugar. Calyx means coat, so glycocalyx is a sugar coat. So bacteria have a sugar coat. There are two kinds. Okay, the type number one is called the slime layer. Like the name sort of hints towards, it is slimy. It is a sugary coat that is loosely attached to the cell. So here we have a bacterial cell, and here's this gooey, slimy substance that's not really tightly attached. Now, why do bacteria make this kind of slime layer? Glad you asked. So it will prevent dehydration. Bacterial cells, like all cells, do not want to be dehydrated. And it also adds a protective layer to the bacterial cell. Now, let's say this particular bacterial cell was attached here. Okay. And all right, if you slept with your mouth open, well, then these bacteria could dry out. Now, one of the things bacteria also do is they talk to one another, and if there are enough of them together, they start to communicate with one another. This is something called quorum sensing. No, you don't need to know this, but they talk to each other and they say, okay, is anybody else here? Oh, there are. Okay, if there's enough of us, why don't we just make enough of a slime layer and it'll produce what we call a biofilm. Really, dentists spend most of their careers trying to prevent and remove biofilm on your teeth. Biofilm. Are biofilms only on teeth? Oh, no, no, no. Biofilms, you can find those in this, when you think about your sink and how it can get slimy if you don't clean it, the bowl of your toilet, in tubes, industrial equipment, unfortunately also on implants and on organs. Yes, organs inside the body. Yes, biofilms form there. By the way, bacteria hiding in a biofilm can be about 1,000 times harder to kill. So, if a bacterial cell gets inside your body and it attaches to an organ, like maybe your lungs, and enough of them are there that they form a biofilm, it is very hard to kill them after they have manufactured and established a biofilm. This is true also if they formed a biofilm in your toilet. They are 1,000 times harder to kill with just adding some toilet bowl cleaner, so you got to get in there and scrub. Okay, these are some bacteria. This is Pseudomonas aeruginosa. You can tell that, okay, this is the slimy area, the biofilm. Here are the bacteria, and they're starting to secrete more biofilm. Pseudomonas aeruginosa can infect your lungs and are very, very, very difficult to treat, especially in the lungs of a patient that has cystic fibrosis. Okay? 1,000 times harder to kill. In the laboratory, it's too bad you guys don't have me for a lab, because we would grow Pseudomonas. You can get it from a faucet if you turned on your shower. Pseudomonas is coming through there. But to grow them up in a pure culture in the laboratory, you can really see how these little stinkers make a really nasty biofilm. All right, what's that second glycocalyx or sugar coat? That one is the capsule. Now, the capsule is tightly attached to the cell, and it protects the cell. So this is very important to a lot of pathogenic bacteria, and it allows them to be stronger or more virulent. In what way? Well, if the bacterial cell has a nice sugary coat surrounding it, then it protects it from chemicals like cleaners, but also it protects them from immune cells. It hides their ID tags from the immune response, makes them very hard to be identified in your body. So they enter, your body doesn't see them because they're all hidden. Also, they are really hard to capture because they're slippery, literally. Phagocytes have a really hard time capturing bacteria that have a glycocalyx capsule. Here's an example. There's a bacterial organism named Haemophilus influenza. You might know it as Hib because you get a Hib vaccine. This means it's Haemophilus influenza B. Now, there are actually two forms of Haemophilus influenza, and you probably have one in the back of your throat nasopharynx right now. If you're thinking, no, well, it probably isn't making a capsule because if the bacterial cell is not making a capsule, it's not very virulent. It's like, it's probably fine. Your immune system will keep it from causing any trouble. But the Haemophilus influenza type B makes a capsule and is very virulent. How virulent? I'm glad you asked, actually. First of all, let me show you. Here is Haemophilus influenza not making a capsule very well. Here it is making a capsule very well. See this clear zone? Looks like a little halo. Yeah, check it out. So, okay, so how virulent? This one gets into the body and is not seen. And even if it is, it can't be captured and it can cause meningitis. So meningitis is an inflamed covering of your brain, or what we call the meninges. Remember in anatomy, the covering of your brain is called the meninges. If it becomes inflamed, that's meningitis. And this is very, 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 very serious. Okay? All right. How about another structure? Okay, these are called endospores. I had to add, in the picture, I had to add my own little structure here because, yeah, the cartoon that I got didn't have that. Now, some bacteria, not all, produce something called an endospore. Now, you might be thinking, oh, is this like a reproductive spore? Like, oh, fungal spores and things like that, one living creature produces a whole bunch of spores and they go out and they spread. No, it has nothing to do with reproduction. Nothing to do with reproduction. This is for survival. Okay? It's when the cell needs to go into hibernation. Okay, so let's talk about this. Here's a bacterial cell making an endospore. If you're thinking that, yeah, that little clear zone. It's clear because stains do not penetrate this endospore, so it looks clear. The stain covered everything else, but not where the endospore is forming. So endospores are the toughest form of living thing. Well, it's the toughest living form that we know of. So, I mean, nothing penetrates. Not even a stain. How about disinfectants? Antibiotics? Nope. You got to catch them when they're in the regular stage. We'll talk about that in a little bit. Endospores are essentially immortal. We found some that are hundreds and thousands of years old. Yeah. By the way, when we sterilize, nothing is considered a sterilizer unless it can kill endospores. So, we have a little thing here that heats up, like your surgical instruments. Will it kill an endospore? Yes, then it's a sterilizer. No, then it's not sterilizing. For something to be sterilized, these things have to be destroyed, which is tricky. Now, only some bacteria make these and only in response to a threat. Okay? So, what bacterial types make endospores? Typically, they belong to the genus Bacillus or Clostridium. So the first name Bacillus or the first name Clostridium, we've heard those names before. You'll hear a lot more. And some of you are like, I know this one. Good. Okay. So I'm going to go through 3 phases of an endospore forming bacterium. Understand that if these bacteria are happy, they look like any other kind of bacterial cell. So here we have, okay, some bacterial cells, and they grow, they eat, num 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 num, and they produce ATP cell energy. Woohoo! They replicate. They do everything a normal cell does. They're just, nothing different about those. So down below, and it's circled in red. Here's a number 1. And this is Clostridium difficile, and he's just being normal Clostridium difficile. Okay, that one there. This is the vegetative phase. So they're in this phase when there's enough food, it's a good, healthy, safe environment. Whatever gas they like, oxygen or something else, is there, nothing poisonous out there. So the cell is normal, it's undergoing metabolism and reproduction. Now, if the bacterial cell is experiencing some kind of environmental threat, such as a lack of food, which is usually what does it, or maybe the temperatures are high, or maybe there's radiation like UV light, or in some cases, believe it or not, some of these bacteria do not tolerate oxygen. Oxygen would kill them. Clostridium, for example, would die in the presence of oxygen. So if they detect this, they're thinking, oh dear, okay, we need to go into the endospore stage of our life cycle. Okay, so here we have, and I know it's very busy. No, you do not have to memorize all of these stages at all. Okay, but here's what's going on. So you do need to know that. The bacterial cell starts to make a tough spore coat, and they're going to form it around their most important uh, uh, components. So they're going to have what? What would they need in there? Well, when we're talking cells, they got to have DNA in there. Ribosomes, we'll talk about those. They make proteins, any essential enzymes, they got to get in there. So that's what they're going to start throwing into the endospore, and then they're going to form a very, very tough coat around those. So down here, number 3, you can see here's a bacterial cell that's starting to feel the threat. And here's the endospore being formed, okay? Now, eventually the whole cell will break down. So here we have the cell breaking down, and what is left is the endospore here. I'm going to move down to number 2. So can you see this? It's just a circle that really doesn't show up very well because the cell is gone. It has disintegrated, and all that is left is this free endospore that is seriously tough. Okay? And it can live in the environment indefinitely, or at least until it enters a healthy environment again, something that it likes. So, like I said, this is not a reproductive spore. This spore has no metabolism. It goes into hibernation completely. All right, so what happens when it lands in a good environment again? Well, that is called germination. The spore has landed in a good, happy environment, and there it sprouts into the normal vegetative cell again. So this is germination. So this would be, again, we're back to illustration number 1 down here. Okay, got that? Some of you might have an endospore former. You might want to talk about this in your life cycle. Just saying. So again, what bacterial types make endospores? They typically belong to the gram-positive members of the genus Bacillus or Clostridium. What would that include? Well, first of all, Bacillus, this means SP for specific epithet, so some kind of bacillus. Bacillus anthracis, for example, right? Bacillus, that means it makes endospores, right? Bacillus anthracis, anthrax. Some things are maybe clicking in your head about endospores and anthrax and okay. Other species belong to the genus Clostridium, such as Clostridium tetani. The tetanus spore, so when you think you get a tetanus shot, okay, and you always worry that, okay, that there's some rusty nail that gave you tetanus. You don't need a rusty nail. All you need is some dirt that gets through your tissue. So you got a cut or an abrasion, and a Clostridium tetani endospore can be floating around in the dirt for how long? Indefinitely, right? Enters the tissue and germinates, causes tetanus and lockjaw and death, by the way, unless you can treat it quickly. We'll talk about that. Clostridium botulinum causes botulism. Clostridium difficile. You know it is C. diff, causes C. diff membranous colitis. Most of you know exactly what that is, too. Okay? All of these endospore formers. Okay? If you see the first name Bacillus or Clostridium on a test, think endospores. And you might even think it's gram-positive, too. Okay? Okay. Back to anthrax. Remember the powder? Went through the mail after 9/11? The powder went to the New York Post, to Tom Brokaw of NBC TV, to Senator Daschle, and, well, it kind of got around. But, gee, how do you suppose they sterilized those areas exposed to anthrax endospores? What do you think they did? Toughest things to kill. Well, in some areas, they had to disassemble an entire room and autoclave every part of that room and every piece of equipment. Yeah, those endospores were the anthrax powder. Okay, so they're so tough. How tough are they? That sounds like a joke. Endospores, we know, can survive in alcohol without a problem. We can grow them up in alcohol. We found some that are 100 years old. We plated them in the laboratory. They grew up just fine. Now, I know what you're thinking. But for those of you that are savvy and you've ever read the ingredients here, ethyl alcohol, 70%. What's the rest? Water and glycerin, that's kind of goo. The active ingredient. What is she saying? What is she telling us? Oh no! So is this going to work at all? Da, 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 da. Yeah, that was, okay. I know, we don't want to hear this, do we? Yeah. Nope. Won't work in the slightest. Purell alcohol-based hand sanitizers will not work on endospores at all, ever, 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 ever. Will this work? Yes! You can't sterilize your hands, you guys. You wouldn't survive. However, with soap, it breaks up the dead skin cells that might harbor endospores, breaks it up, washes it all down the drain. Safely for you, safely for your patient, safely for your family. Soap and water. Not sexy, but very important. What if your patient in the hospital room has C. diff? I know, you're not believing me, are you? Or maybe you do. Maybe you do remember somebody, the infectious control nurse, those wonderful heroes. You probably remember them telling you you cannot use a hand sanitizer alcohol-based hand sanitizer around a patient with C. diff. Okay. By the way, my brother died because of C. diff and Pseudomonas aeruginosa, neither of which he had when he went into the hospital. So where did it come from? Yeah. Okay. So let's talk about plasma or cell membranes, okay? Now, all cells, is it a cell? Yes. Okay. All cells have to have a cell or plasma membrane. All of them. Okay? Do our cells have to have them? Yes. Plant cells? Fungal cells? Yeah. Malaria cells, yes. Okay, if it's a cell, it has a cell membrane. Now, if you remember your chemistry, and you remember that that cell membranes, plasma membranes, are made up of phospholipid bilayers with proteins inserted in there, and it makes a semi-permeable barrier for the cell. Okay, so again, you have the phospholipid bilayer. You have these proteins in purple. You sometimes have glycoproteins sticking out of those as well. And again, it's semi-permeable. It lets some things through, but not everything. Understand this is not a cell wall structure at all. Not a cell wall. If you're thinking, okay, I got it, back off. Trust me, because when I talk about bacterial cell walls, a lot of students get mixed up. Not just you guys, all of my students. They always say cell, or there's always a student on an exam who says cell membrane when they mean cell wall, and they lose those points. Okay? And trust me, I will have questions that will distinguish cell walls versus cell membranes. So do not mix them up because, okay, when it comes to membranes, okay, prokaryotic membranes, just like all cell membranes, are pretty much the same in that they have simple and active diffusion that regulates the movement of gases and nutrients and waste products in and outside of the cell. That's what they do. It regulates what moves in and out of the cell. If a membrane, the semi-permeable membrane, is disrupted, the cell dies. I'm not going to talk about the kinds of drugs that disrupt plasma membranes until we talk about fungal organisms and antifungal drugs. Okay, but we will later on. Okay. Speaking of drugs, let's go into special morphological structures, that means structures of the cell, and processes of the cell that are targeted by antibiotics or antimicrobials. Okay? So is there something special about bacteria that we can target? Understand, an antibiotic is a poison. You're taking these orally or even through IV. The reason they don't poison you is because they target bacterial cells and hopefully not your cells. So, can we find some special targets for these antibiotic poisons? Oh, yeah. So, here we go. So, first of all, we're going to talk about the cell wall of bacteria. Most bacteria, not all, but most do have a cell wall. And the major component is something called peptidoglycan. Okay? So, cell wall, peptidoglycan. Notice it's not a cell membrane, cell wall. Okay. The cell wall is a very strong, rigid structure that prevents cell lysis. That's what it does. It's strong, but it's like scaffolding. It doesn't keep things from moving through it. It just keeps the cell from maybe blowing up. If you remember learning about a hypotonic solution and water can rush into a cell and the cell expands until, okay, well, a cell wall will keep from happening. Okay? Now, in bacteria, the cell wall is made up of peptidoglycan. There's that word again. Do I have to know that word? You sure do. That one, absolutely. Peptidoglycan is very unique, even though not all bacteria have a cell wall, and not all bacteria have peptidoglycan, but it is uniquely bacterial. Oh, can we use this information to destroy bacteria? Yes, we can. So the peptidoglycan structure distinguishes two main types of bacteria, something we call the gram-positive bacteria, and then there are the gram-negative bacteria. Now, if you've been doing your research, and I'm sure you have probably come upon these words. It's usually the first thing an author says about the bacterial organism is, well, this is a gram-positive, whatever. Oh, this is a gram-negative, whatever. Okay, so we're going to talk about these. So what is a gram-positive and a gram-negative? You know, what is this gram stuff? Where did this come from? Well, we're going to talk about that. And it's all about the bacterial cell wall and structure. First of all, okay, so Gram, who is this guy? What is this? Gram, the name Gram comes from a scientist who is Dutch. His name is Hans Christian Gram. Ah, got it. Now, Dr. Graham was working with a lot of chemicals and stains in order to see different kinds of bacteria. Now, he realized that certain chemical stains, so they were chemicals that had a color, they reacted with some bacteria differently than others, and that allowed him and us to differentiate these two types of bacteria. Okay, so really, the Gram stain that is done allows all of us to understand the cell wall and cell structure. Now, let's go through the procedure really quickly. It's too bad that we can't do this in the lab because this is not very meaningful when you hear about it. It's like, okay, whatever, moving on. But still, I have to go through it just to make some sense of it. So bear with me, okay. All right, so first of all, we always add, when doing differential stains, we always add a primary stain. What Hans Christian Gram did was he added something called crystal violet. So it's purple color, right? It's a purple color. And all of the bacteria would just kind of hold on to this purple color, and it would hold on to all types of bacterial cells at this particular time. And he'd wash off anything that was extra. But then what he would do is he would add a chemical known as iodine, which was a mordant. This iodine would cause the crystals in crystal violet to crystallize. Okay, so again, everybody's still looking purple. But then he would use what's called a decolorizer. He noticed that some bacteria pretty much disappeared. And only some would retain that purple color, the crystal violet. Now, another scientist, and I won't go into that, but he wanted to see the bacteria that would disappear, so he added a secondary stain, and we call that the counter stain. And he said, well, let's add a different color, and so there's something called red safranin, and he added that. So now we can see the other cells as well. Now keep in mind, all of these cells were exposed to the exact same chemicals and processes. Okay, but some remained purple, and some lost the purple color and became red. What is the significance here? Oh, sorry, before I leave. So here are some Staphylococcus aureus cells, and here are some E. coli cells. Again, they all went through the Gram stain process together. Some turned purple, some turned red. Okay, so let's take a look at really what the Gram stain is telling us about the morphology of the bacterial cells. The structures. Okay, that's what this Gram stain is really telling us. And that's why different stains react differently to different kinds of bacteria. All right, stay with me. So the bacteria that we call the gram-positive types of bacteria, they look like this. They have a lot of layers of peptidoglycan in their cell wall. The crystals of crystal violet, okay, well, first of all, the crystal violet stain gets in here, and then you add the mordant, and the stain crystallizes. The chemicals crystallize, and the crystal violet gets jammed in these multiple layers of peptidoglycan, and so they don't wash out. Okay, the cell then remains purple. Even if you add the red safranin, yeah, the purple color still dominates you can still add the red safranin. It's kind of a mild red color. It actually is pink, but they're still going to look purple. So again, this is all the outside of the cell wall. Okay? or the outside of the cell, sorry, in the cell wall. The gram-negatives, now these are different. What they have is an outer protective membrane as well as an inner membrane. What? Okay, look at the bacterial cell here. There's two membranes here. And the peptidoglycan is very thin. There's only a few layers of it. So if you add the crystal violet, a lot of it just kind of gets stuck in the upper protective membrane. And even if any of it gets down here, there's not a lot of peptidoglycan, okay? So when you decolorize, well, everything washes clear. You add the red saffron in, and now they look red. Otherwise, they look completely clean or clear. So crystal violet, the crystals don't get in here really very well. They just attach to the the very outer portion of the bacterial cell. When you decolorize, it just washes right off. And then we just use that second color in order to see them. And there we are. So up here, this is Staphylococcus aureus, by the way. Look how they are in a staphylo arrangement, and they are coccus-shaped. And then we have E. coli, which is pretty much a single bacillus, right? Gram-positive, gram-negative. So Staphylococcus aureus is a gram-positive bacterium. E. coli is a gram-negative. I'm going to have to bring in another term here, and this is the cell envelope. So what is the cell envelope? This is a term we use to talk about anything surrounding the bacterial cell. So as you can, from the last slide and from this slide, you can see that, well, okay, the outer portion of the cells are very different. So they have different morphological characteristics. So, for example, when we talk about the gram-positive bacteria, they have one membrane and peptidoglycan in many layers. The gram-negatives have two membranes and a few layers of peptidoglycan. So all of this surrounds their respective bacterial cells. So we call this all just the envelope. The envelope, anything surrounding the bacterial cells. So this includes all membranes, cell wall materials, and anything in between. And again, why do we do the gram stain? Because the gram stain tells us what they look like. Why do we care? I'll give you a why do we care moment coming up. Okay, first of all, let's talk about that peptidoglycan. So again, you are here. Here's your bacterial cell. There is a membrane and lots of layers of peptidoglycan here. So we're going to blow that up. Here's your normal plasma membrane. Here are the many layers of peptidoglycan. So the peptidoglycan, if you remember your chemistry, what is this made of? Well, peptide, oops, that sounds like proteins kind of, right? Like amino acid chains? So here we have these chains of amino acids. So these are peptide chains. And then glycan, golly, that sounds like a sugar. You're right, and sugar bars. So peptidoglycan means that the cell wall is made up of peptide chains and sugar bars. So the peptide chains are 4 amino acids and they're linked together. And then you have these long sugar bars here. And the sugar bars are made up of chemicals known as NAG and NAM. You're like, what's that? N-acetylglucosamine and acetylmuramic acid. If we look up here, the little pink circles, it's a disaccharide. You have pink circles and blue circles. And that goes NAG and NAM and NAG and NAM and NAG and NAM. They're all hooked together, okay, to make sugar bars. So you only need to know them as NAM and NAG, okay? And then, okay, so how do we link the sugar bars together? Well, that's where we do the peptide chains, okay? And they have to be linked together if we're going to have a nice, strong cell wall. Is that really important? Yes, it is very important. At least to the bacterial cell it is. Okay, so gram-positive cell walls have lots of outer layers of peptidoglycan just sticking out there like a target, really, for chemicals and things. So the gram-positive cell wall has a thick, many-layered area of peptidoglycan outside the one-cell plasma membrane. Again, gram-positives. All right, so how about some more details? All right, because there is more to this. Knowing that your cell is gram-positive not only tells you that there's a lot of layers of peptidoglycan, but there's also a little chemical known as teichoic acid embedded in here. Now, unfortunately, it doesn't show it in this schematic, but there's these little long strands of teichoic acid kind of sticking out, and these are little identification tags, and they peek out between the peptidoglycan cell wall. So they're attached to the cell wall as well as the plasma membrane. And because they stick out there like an ID tag, they are antigenic. Remember that word antigenic? It generates an immune response. Now, something else about gram-positives. They are most susceptible to penicillins. And what do penicillins do? The penicillins attack the enzymes that make peptidoglycan. Okay, I've lost you. Stop for a moment. Okay, here's the bacterial cell, right? And it's got to have a cell wall. So here's the cell wall. So how does this thing get constructed by the cell? Well, if you remember Mr. Seberg's lesson, most anything that gets done in a cell or even outside of a cell has to be done by an enzyme. In my cell biology class, I've always told students, okay, the answer to almost everything is enzymes. How does a cell make this? Enzymes. Well, how does a cell make that? Enzymes. How do we stop a cell from making something? Attack the enzyme. So there is a little enzyme out here that links together these little peptide chains that is needed to make a strong cell wall. Penicillin attacks that enzyme trying to do its little job out here, linking these chains together. Again, my example of a gram-positive bacterium is Staphylococcus aureus. Okay? All right. How about gram-negatives? Why are they different? Well, again, they have a whole different cell envelope. And their cell wall is there. I mean, they do have peptidoglycan, but it's a thin layer, maybe just a few layers of it, okay? So it's there. So these are gram-negative bacterial cells. So the one unique thing about gram-negative bacterial cells is that they are protected by this outer membrane. It is freakish in the cell world. Okay, look at this illustration again. Here we have a plasma membrane in green. You have the cell wall of peptidoglycan in purple. And then you have an outer membrane. Okay, everybody raise your hand if you've ever learned about a cell that had two outer membranes. Yeah, nobody's raising their hands because you've never heard about this before unless you have studied not just bacteria, but gram-negative bacteria. It is the only cell that has a double layer outer of membranes, okay, in the envelope. So, gosh, that peptidoglycan is pretty protected. In fact, the whole cell must be really protected because membranes are semi-permeable and they regulate what moves in and out. So if there's a double layer of protection, these must be really, really tough cells. Okay. So again, not even the crystal violet got in there, so they'd stain red. So again, do any other cells have two outer membranes? No. No. Is there anything else that's really cool about gram-negative cells other than they're really tough? I'm glad you asked. Okay, let's talk about more aspects of the gram-negative cell and cell envelope. So again, they have an outer membrane, and inside is called a periplasmic space. So again, this is the area between the two membranes. So, and then also inside the periplasmic space, that's where you have the peptidoglycan cell wall layer. That outer membrane is very protective, as I mentioned. Many gram-negative bacteria are not even hurt by penicillin because they have the outer protective freakish membrane out there. So, also, something really important in that outer freakish membrane is something called a lipopolysaccharide. What? Or LPS. And it sticks out here. There are two parts to the lipopolysaccharide. Okay. First of all, the polysaccharide part sticks out like an ID tag. So it's antigenic. It's an antigen. When these bacteria get into your body, they are identified by these LPS sugar-like parts that are coming out. So that's one part of this molecule known as LPS. Now there's an inner part of LPS, and that is the lipid part. And that is an endotoxin. Okay, so we're here. All right, so the outer part is the sugar part. The inner part is the lipid part. So lipopolysaccharide, LPS. And again, are we finding it on this membrane? No. Are we finding it in the peptidoglycan cell wall? No. It's only in this gram-negative outer freakish membrane. So it's an endotoxin, and that doesn't sound good. No, it doesn't. Endotoxins in small amounts will cause fever. In large amounts will cause shock and death. More about toxins later on. Okay, some questions. Let's pause for a question. Which of the following types of bacteria is lacking a cell wall? Gram-positives or gram-negatives or none of the above? That's my cat purring in the background, so ignore that. Right, no, none of the above. They all have a cell wall. They all have peptidoglycan. Which one has more? Gram-positives do. Good, A. And now let's just pause for a moment. I have something to help you remember this. Because you're thinking, I'm going to forget this the minute I turn off her video. Okay. All you have to do is remember some P's. We can stress the gram-positives, and then that will also help you differentiate between the gram-positives and gram-negatives. So remember the P's. Okay, my cat is being obnoxious. All right. So the P's of gram stains. This only applies to gram stains. Only. Gram-positives stain purple because they have many layers of outer peptidoglycan, and they're more susceptible to penicillins. Got it? Gram-positive stain purple because they have the outer layers of peptidoglycan, and they're more susceptible to penicillins. So what about gram-negatives, you ask? Okay, not as much. Okay, gram-negatives, they have the freakish outer membrane. So it's not a gram-positive. They have the outer freakish membrane that makes them red. Okay, and they have the LPS. But at least that helps you distinguish the gram-positives, right? I don't know. I think I need to feed my cat. So let's pause. You go get some caffeine. I'm going to go feed my cat, and we'll come back.